the recording, share the screen. All right, can you see the screen? We are going to cover Lab 6 on diffusion and antibiotics today, and we're also going to start and discuss the unknown project. Oh, good heavens, I've got to get that mailed out to you. All right, any questions about what we're doing? So let's begin with starting about the unknown projects. Under the lab module, I think it's under week 5, but maybe it's week 6, there's a file called unknown instructions, and it will be the introduction for what you're supposed to do. I'm going to send you out your unknown number, and actually, you know, I need to do this. I need to show you where this stuff is, so let me log in there. To the Canvas website. So under week 5 lab, we're doing lab module 6 today, and there's the worksheet to turn in. And then underneath that, I've got the unknown report, and hopefully you've taken a look at the unknown instructions. I'm going to briefly talk about them. When you're doing your unknown project, you should start recording things in your unknown notes. Let me open that. And I won't have it because I'm an instructor, but over on the right you will have the download, and you download multiple copies there. And so each day when you're working on the unknown project, you will write up your notes for that day in the download there. Any question about that? I've got the speed grader so I can look at each person's thing, and I'll bet there's nothing up here because nobody started on this project yet. I won't be grading until the end of the term. Near the end of the term, I'll give you a grade on how well you kept up the unknown notes. But each time you do something, you'll have a download there, and then I can go look at each submission you made, and I think it's defaulting now to the latest download will be the one that will be highlighted and shown. That reminds me, when you put in an assignment, some people have learned that you should put in like the upload to when I'm requesting changes, you should just put it in again. I'll have access to both of those, and let me go to an example of that. Think about this, how to get that. I guess I can't do that because I'll show you somebody's grades, but I will have access to it, and I can look on something. So when I'm asking you to make a change, you should download it in the same place. Some people have started giving me comments. I don't want comments, but if you want to send me a comment, that's fine. But do not attach a document to your comment, because I may not see that document. You should be downloading your documents in the assignment area. Okay? Do not download your new documents in the comments. Alright, anyways, for the unknown notes, you'll be recording information here. The first thing you should do, once I've given you your unknown number, you should download in your unknown notes what your unknown number is. And then, later on in the term, when you ask for a task, I will look for what your unknown number is in your unknown notes, and then get you the test results for the test you're asking for. Okay? Any question about that? So it is important to stay up to date in your unknown notes, and it's important to document what the heck you're doing, and you will be graded on it, although it's only worth 5 points. Okay? Any question about any of that? So that's something that's due on March 12th for 5 points, but you should be able to, I guess I should look into that, make sure you can...
Question, Professor?
I'm sorry?
A question.
Yeah.
Have you sent the numbers to all of us?
No, I told you I haven't done that yet.
Oh, okay.
But on the unknown notes, you should get it January 26th. You could look at that. I need to do that. I have not done that. In fact, I forgot that today was the day that you're starting the unknown project. I knew I was talking about it today, but I didn't remember you're actually starting it. And that's true that I want to get you guys started right away on this project, because some of you, it will take time to get this project done. You do have nearly to the end of the term to get it done. Let me see when the report is due. Oh, shoot, I don't have it here. Let me look down here. Way down in week 11 or 10. It's due March 13th. Okay? The unknown report is due on March 13th. You have time, but some of you, it's going to take a lot of time before you'll find what your unknown bacteria is. And so I want to get you started early. And then it's not my fault, because you've had lots of time. I'm giving you lots of time to work on it. But let's talk about the unknown instructions. And let me, while I'm here, point out that there's obviously the link to the unknown instructions that I'm going to talk about. There's a sample journal which you can use to write up your journal report. Now let me warn you that this sample journal was done by a professor many years ago. It was something like 2005 or 2004, something like that. And back then, we were giving students 2 bacteria, and then they had to streak it out, isolate them, and then purify them, and then use one of them to study for their unknown report. We decided that because this is all going to be done online, you're not going to be coming into the lab to do any of it, that we decided not to give you a mixed culture where you would be streaking it out and then isolating one species and then doing a report on one species. We're going to send you just one species, and you're going to try and identify what that one species is. And this report, meaning the sample, starts out where he's got 2 species, and he's streaking it out for isolating it and then purifying it, and then using one species to write up his report. And you're not going to do that, because I'm only going to give you one species. So the beginning of this you can kind of ignore, because you're not doing that work. But the rest of it you are doing. Any questions about that? All right, that's an example for what your journal should look like. And then you got to use a dichotomous game plan for doing your unknown report, meaning that you can't simply wait to the end and say, give me all of the tests. I want to do all of the tests. You could do that, but you'll be marked down because you're not using a dichotomous game plan. For a dichotomous game plan, you should do one test, which will then be plus or minus, and then when you get your results, you look at what the possibilities are and say that, oh, I don't know, you got the gram stain, and so it's gram positive. And then you do the lactose fermentation test, and it is acid and gas, okay? And then you look at what the possibilities are, and then you decide which test to run next. That's using a dichotomous game plan. If you do not use a dichotomous game plan, you will be marked down on your unknown report. Okay, so you should be using a dichotomous game plan. Obviously, at the beginning, you have to consider all the possibilities. But then, you will do a test like the gram stain that will narrow your possibilities down to either gram positive or gram negative bacteria. Any question about that? And then once you have that, you do a test based on the fact that you have gram positive, for example. That's the dichotomous game plan. All right? Now, the first test you're going to run, I'm going to give to you, and that's because I looked at this and I said, you guys, this will work the way we're proposing to do it, but do you realize that for each student, we're going to have about 20 different contacts? And they're not really emails, because what you're going to be doing is you're going to be writing your notes in your unknown notes, and then you're going to be asking for a test in your unknown notes. So at the end of the day, you submit what work you did and what your notes are and what you're thinking and what your possibilities of are, meaning the possible species. And then at the end of that, you say, I would like to do this test next. And then I'll come and take a look at it and see that you need to do that test. I'll look to see how long that test runs, and then I'll get you incubating it online, meaning you're not actually going to incubate it, I'm not going to incubate it, but it's going to incubate for the proper amount of time online. And how you can find that out is come down here for the test, and then let's say you want to do the bioesculin test. You click on this and read up on it, and you'll find how long the test is. I think bioesculin runs for 2 days. I'd have to look that up to be certain. Most of the tests run for 2 days. The catalase test is, you can get the results, assuming you have fresh bacteria cells, you can get the results in the same day that you ask for it. But most tests are not that quick. Most of them have to be incubated for 2 days. One of the tests has to be ran for a week, the gelatin hydrolysis test. Okay? And I decided that that one splits the bacteria up pretty nice. So does the gram stain. Let me show you where that is. The gram stain. And this also tells you the cell shape and the cell arrangement. Realize that when I send you the gram stain, you only get the gram results. To get the cell shape and the cell arrangement, you have to do another test. My point is that this test, this project would take, I figured, 20 emails or messages per student. And I said, that's just way too much for any one project because there's 20 students, and if each of them takes about 20 messages, that'll be way too much communication between me and each student. And I wanted to narrow that down. And I thought of a way of doing it, and that is, instead of sending out the first 3 tests to each student, I'm going to pick 3 tests and send everybody in one email 3 tests. So instead of sending out, if there are 20 students, and this class has gotten smaller than that, instead of sending out 3 times 20, which is 60 messages, I'm only going to send out 1. And that greatly reduces the messages between me and the students. And so your first tests are going to be decided by me, and I will send you them all together in one email to everybody, not just one student, but to all 20 students. And you're going to get 3 tests, and they're going to be the gram stain, the gelatin hydrolysis, because that one takes a week, and I think it's lactose fermentation. I think it's those 3 tests. And what you're going to do is, I'm going to tell you that you're going to start the gelatin today, if I get your unknown numbers sent to you today, because it takes a week for this test to run. You're going to start the gram stain on Sunday, and let me think about that. That's not right. You're going to start the lactose, yeah, you're going to start the gram stain on Sunday, and then you'll start the lactose fermentation test on Monday. And then on Tuesday, I can send everybody the results to all 3 of those tests, because the gelatin hydrolysis will run for a week. The gram stain will be grown. And for the gram stain, remember, you have to have fresh growth, so you have to grow it out 2 days before you start on it. And you started it on Sunday, but you never looked at the slide until Monday or Tuesday, and you got your results on Tuesday. And then you're starting the lactose fermentation test on Monday, and that one ideally should run from 18 hours to at most 48 hours. Okay? And that's why you're starting that one on Monday. And then you'll get the results on Tuesday for all 3 of those tests. That's the only time you will get 3 tests on one day. But remember, you're not running 3 tests on one day, because you're starting the gelatin test today, assuming I get your unknown number to you today. And you're going to be starting the gram stain on Sunday and the lactose fermentation on Monday. So you will be doing a dichotomous game plan. Okay? And I'm doing it this way just to reduce the number of contacts between me and the students. And all of these contacts are going to be done in the unknown notes. All right? Any questions? Once again, in the unknown notes, do not put your comments in the comments sections. I will put my comments there. You want to put your comments in the body of your work, meaning you put it in a document and then submit the document. You'll have a button for that over here. Okay? And then at the end of that document, write which test you'd like to do next. Okay? All right, any questions about any of that? Everybody's good? All right, so that's how you do it. And there is a grading rubric for how the unknown report will be graded. And I suggest you don't look at that until at least halfway through the term. You do need to look at it before you write up your unknown journal report and then submit the journal report. This link here is the possible unknown species. So you can click on this list. Let's go ahead and do that. I think there's about 20 unknown gram-positive species and 20 unknown gram-negative species. I'm opening it this way because it'll be a larger print. This is important because it shows you the possibilities. The top is the gram-negative bacteria. The bottom are the gram-positive bacteria. And then it has the species over here. And then the different tests that you can run on the top up here. And it goes over to here. And so on one of the tests, you will be given the gram test. You will immediately narrow to either the gram-negative or the gram-positive bacteria. If you look at the gelatin results here, that's another test. About half are plus, half negative. Okay. Down here, it tells you some helpful tips for like running the gram stain. The cell arrangement, and pay attention to that tip. Students every turn get that one wrong. Gelatin, tips about 6.5% sodium chloride, 7% sodium chloride, urease, etc. Then how do you read this? Obviously plus, meaning positive results, minus negative results. V means variable results. That means that a Micrococcus varians, let's see what that test is. I should use this one, it's easier to read. Enterobacter cloacae, if you grow it in 6.5% sodium chloride broth, it'll be a variable results. Sometimes it'll be positive, other times it'll be negative. So you have to record this variable as both positive and negative. Okay? So that's not good news if you get a variable, but on the chart, I'll never give you a variable like you did the 6.5% sodium chloride. I will give you either a negative or a positive result. And if you think you might have Enterobacter cloacae, regardless if I give you a negative or a positive, it'll be a match. Everyone clear on that? Not determined also means the same as a positive or a negative. So like for example, if you're running hemolysis and your unknown is a gram-negative, I will give you the results, but it's going to be meaningless for a gram-negative because none of them are gram-positive or gram-negative. Okay? Any question about that? The asterisk telling you that generally the shape of the bacteria is Cloacae, E. aerogenes. Generally they're bacilli, but they're short bacilli. And L. lactis is generally a coccus. But sometimes these three species can be Coccobacillus in shape. And let's see where that is. right there. And where are those others? Right there and right there. All right. Any questions about the unknown chart? If you have questions about it, you can ask me later. All right, I think that's everything here. Let's start talking about the unknown project. You'll have to write up your lab report and do the project. Everything you'll be doing is online. Are there any questions about that? You're not going to go into the lab and do any of these tests. And you're not going to go into the test and get right into the lab and get test results. Everything's going to be done online. And that's because of COVID-19. You need to write notes for every day you're doing something. It doesn't have to be daily. For some of you, it will be close to daily. But whenever you're doing something, you should write up your notes. You should do those on the unknown notes pages. And then if I have any questions about your unknown report, I can go look in the unknown notes pages. Both will be graded. The unknown report project will be worth, I don't think it's 100 points anymore. Let me check on that. In the syllabus, I think that's worth 75 points. Yeah, the unknown report is worth 75 points. This is an old copy I have on my computer, and hopefully I've updated your thing, because that's a little bit misleading if it's saying 100 points. If you have something that says 100 points, let me know and I'll correct that. But I think I've already updated that. The unknown report must be typewritten, and you have to make it electronic copy and then submit it in the Canvas website. Hard copies cannot be turned in because we're not ever going to get together. The report should be written using 12-point font. As you can see, I wear glasses and have trouble reading if the font is less than that. You should not double space. You should write the report in bullet points, but it won't mark you down if you write in essay style. You can submit drawings, photos, and tables. They are very helpful for reading and understanding your lab report. They are not required, except there is one section. Let me see where I've got that. The grading criteria, we'll get to that. You do need to have a table. I'm not finding that now. You do need to have a table at the end saying what tests you use to, oh, it's right here. Never mind, that's why I can't find it. It's right in front of me. You need to have a table or a figure saying what tests you used to identify your unknown. Like this student ended up with Micrococcus varians, and they got it because it was a gram-positive. These are the possibilities after it's gram-positive. You don't need to say what the possibilities are before that, because that's all the species before that. You just in your report say, I considered all the species, and after the gram stain realized I only had either the gram-positive or the gram-negative, and so this is the possibilities. I then did the Cadley's test, and it was positive, so which is right there. Cadley's positive, which means that it's these 4 possibilities. I then did the PR mannitol, and it was these possibilities. And then did the PR glucose, and it was these 2 possibilities. I then did the citrase test, and it was positive. And only Micrococcus varians could fit the possibilities. So my unknown is Micrococcus varians. Okay. You could do one more test, which is a confirmation test that your unknown is Micrococcus varians. I will allow you to do one confirmation test after you think you've identified your unknown. Okay? Any question about any of that? What happens if you do more than one confirmation test after you've identified your unknown? Come on, somebody can guess. All right, somebody will have to do it because nobody's guessing.
I would guess we will get extra points.
No, you won't get extra points. You'll get penalized if you do more than one confirmation test.
Oh, come on.
All right, let's go in here. Each report date must begin with the date. You don't really need the time because on one day you will only be having generally one report, one test per day. If for some reason you do have more than one report per day, then you should also put the time. And I'm not terribly worried about that because the unknown notes page will put the date and the time on automatically. But when you're writing up your lab report, you should have each report starting with the day. You don't need to put the step-by-step details of the test. I know what the tests are. You just need to tell me which test you ran, why you ran that test, what you were hoping to see from that test, what the possibilities were, and then what the test results were, and then what the possibilities are after that test result. The thoughts and words must be your own. If you're not using your own words, thoughts and words, then you should put it in quotes and say where you got it from. Otherwise, it is plagiarism. And if you plagiarize anyone, you'll get a 0 on this report. And meaning you don't have it in quotes, and you know what I mean. You must have a table or a figure or a flow chart outlining the process you use to identify your unknown organism. You must have at the very end a microorganism paragraph, meaning once you've outlined and identified the species, at the very end of the report you should have a paragraph discussing that species, like where it's found, what it does, etc., like that. And that should be only one paragraph at most, two. You should have a literature cited section. So especially in this paragraph at the end, you should have an in-text citation for where you got some of that information, and then a literature cited section for where you got the information for your lab report. I've got some information about plagiarism there. And it says that if you plagiarize and I find about it, there will be a 5% penalty minimum, up to 75 points. That should be 75 points. The grading criteria, I'll let you read about that. I'm not going to say that other than that you should have logic for what test you ran. You should have completeness of the report. You should be organized, clear, neat. You should follow a proper laboratory technique, and that'll be pretty easy because everything you're going to be doing is done online. All right, that's it. You can ignore the bottom here. This is one way to isolate a mixed culture. because normally we give students a mixed culture and then they have to isolate it and get a pure culture. So you can ignore this page and this page too. Although this page you can look at and it show you how to follow a dichotomous game plan. All right, any questions about the report and about the instructions for writing up your report? All right, if you have any questions, email me. All right, let's move on to Lab 6. Disk diffusion. We're going to be evaluating disinfectants and antiseptics, sort of, and mostly evaluating antibiotic effectiveness. There's some pre-reading that should be done. The learning objectives: be able to identify the type of a chemical reagent and the active ingredient used in the various disinfectants and antiseptics, evaluate the effectiveness of different disinfectants and antiseptics using the disk diffusion method, which, like I said, we're not really going to be studying this, but it's pretty obvious how this would work from what we're going to be doing. We will be working at the antibiotics and be able to determine the mode of action of the various antibiotics, evaluate the effectiveness of various antibiotics using the disk diffusion method. Generally speaking, the larger the zone of inhibition around an antibiotic, the greater the effectiveness, but it's a little more complicated than that. That's a simple way of looking at it. The greater the zone of inhibition, the better the antibiotic. But like I said, it's a little more complicated than that. We'll talk about the exact way at the end of this. List the different variables that influence antibiotic disk diffusion susceptibility testing. Be able to define the terms: sterilization, disinfection, disinfectant, antiseptic, bactericidal, bacteriostatic, antibiotic, what's a spread plate, and zone of inhibition. Zone of inhibition, I'll talk about since I discussed it, is when you put an antibiotic disc on a plate that's been coated with a lawn of bacteria and then allow the bacteria to grow. If the antibiotic is effective, there will be a zone of inhibition where the bacteria did not grow around. I'm trying to show that. Let me get a different color. So this is the disc with the antibiotic, and this is the zone of inhibition where the bacteria are not growing around the disc of antibiotic. Any question about any of that? All right. Sterilization means the elimination of all viable organisms, including endospores. In the lab, if you were normally going into a normal lab at Clark College, we will give you sterile media and it would be sterilized in the autoclave. Disinfection refers to the destruction of vegetative cells, not necessarily endospores, usually with a chemical or a physical agent. Like when you use bleach on your toilet, you are disinfecting the toilet with the bleach. Disinfectants are chemical agents applied to an inanimate object. If, on the other hand, you were putting the bleach on a cat, we wouldn't call it a disinfectant, we would call it an antiseptic, a chemical agent applied to living tissues. Generally, antiseptics are less toxic and less caustic than disinfectants, which is why we don't normally use bleach on the cat, because the bleach would be caustic to the cat. However, I don't know if this is still the case, when you had necrotizing fasciitis more commonly called the flesh-eating bacteria. One of the treatment methods for treating that is to put a cloth wrapped in bleach around the infected area. Okay, so sometimes bleach can be used as an antiseptic. All right, any question about that? To evaluate the effectiveness of a chemical agent, the surface of a nutrient agar plate is inoculated with a particular bacterium. Paper discs saturated with different chemical agents are then applied to the surface of the agar. The chemical agent diffuses into the agar, establishing a concentration gradient around the disc. You then incubate the plate, generally overnight, and the bacterial growth will grow on the plate, and there will be a growth pattern around the disc that's observed. If there's a zone of inhibition, an area of no bacterial growth around the disc, the chemical agent is assumed to be at least a little effective for that bacterium. Conversely, if there's no zone of inhibition, the chemical is assumed to be ineffective. Any questions about any of that? And this is showing you a table of the different chemical agents and their general action. I'm not likely to ask you about that, except for maybe near the end of the term where we'll talk a little bit more about this, meaning I'm not going to quiz you on this table, at least this part, the general action in the lab. The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic or the antibiotic. There are various other factors that affect the zone size, including the concentration of the chemical, the molecular weight of the agent, and the density of the bacterial inoculum. Okay? So, if you have a very large molecular weight chemical agent, and it's effective against a bacteria, we expect the zone of inhibition to be small, even though the chemical is very effective at killing that bacterium. And why is the zone of inhibition small? Come on, I just said it. All right, you guys are being too slow here. I don't want to take a lot of time discussing the lab. I want to let you have time to work on the lab while I'm still here. Because the molecular weight of the object was big, it's going to diffuse less in the agar plate. And that is why the zone of inhibition was small, even though the agent was very effective against that bacteria. So do you understand how the zone of inhibition is only partially determined by how effective the agent is on killing or controlling the growth of that organism? There are other variables that determine the size of the zone of inhibition. Everyone got that? Well, no comment. I'm going to assume everybody got that. All right. Evaluation of the antibiotic effectiveness. Antibiotics are categorized as bactericidal if they kill bacteria. They are categorized as bacteriostatic if they inhibit the growth of bacteria. When bacteriostatic antibiotics are used, we, meaning doctors, rely on the immune system to assist in the elimination of the infection. because a bacteriostatic antibiotic will not kill the bacteria. Is that clear?
Yes.
All right. When you look at a zone of inhibition, how can you determine whether it is bactericidal or whether the zone of inhibition is bacteriostatic on an agar plate? Well, while you guys are thinking, I'm going to get a snack. I still haven't heard anything. Nobody even has a guess? I mean, you don't have to be right.
Wouldn't you see the bacteria dying off if it was bactericidal?
Could that be done? No. When you look at bacteria under the microscope, you cannot tell whether they are alive or whether they are dead. They'll still be there and you can see them. Now, maybe after a day or two, the bacteria will slowly disappear because they'll start disintegrating. But I don't think anyone wants to be looking under the microscope for over a day. When you look at a plate that's showing you a zone of inhibition, you cannot tell whether the antibiotic was bactericidal or bacteriostatic. What you would have to do is streak the region where there's a zone of inhibition and put it on another agar plate that has no antibiotics. If you get growth on that plate, that means the bacteria is bacteriostatic. If you don't get any growth on that plate, it means the antibiotic is bactericidal. Any questions? There is another way you could determine it, and that is you could take off the disc containing the antibiotic and then re-incubate the plate. If the bacteria grow within the zone of inhibition, the antibiotic was bacteriostatic. If the bacteria do not grow in the zone of inhibition, then it's bactericidal. All right, so how people use plates, agar plates, to determine the efficiency of an antibiotic for killing a specific bacteria is called the Kirby-Bayer disc diffusion test, and it is the most common method for testing an antibiotic. The results of such testing help physicians in choosing which antibiotics to use when treating a sick patient. The bacteria, like the patient has an infection, and the doctor can grab the infection from the patient, grow it up, and then plate it as a lawn. We'll talk about how to do that in a minute. And then put antibiotic discs down on the plate, and then find an antibiotic that is very effective against that bacteria. The reason why the Kirby-Bayer disc diffusion test works so well at giving the doctor the results for which antibiotic will work against that bacteria is that the antibiotic diffuses out of the disc through the agar, creating the zone of inhibition. And it is true that the smaller antibiotics will diffuse better than the larger antibiotics, but when we give an antibiotic to a patient, the antibiotics are carried by the bloodstream to where the site of the infection is, and then the antibiotic diffuses out of the bloodstream into the tissue, which is very similar to the antibiotic diffusing from the disc through the agar plate. So that the Kirby-Bauer test is very similar to the way the antibiotics work in a patient. And that's why the Kirby-Bauer test, this diffusion test, is so widely used on antibiotics to find out which is a good one to work on an infection. When we're using this test to determine a disinfectant and which disinfectant is the best to work on a bacteria, why is this a poor test to use to determine which disinfectant is the best to work for that specific bacteria?
Probably because you can have multiple different antibiotics against one bacteria.
Okay, we're talking about disinfectants in this case being in the disc, not antibiotics. And then the disinfectant diffuses out of the disc and creates a zone of inhibition. Okay? And so why is this test not such a good test for testing different disinfectants?
Well, that way, because the disinfectant would just spread across the whole thing.
Yeah, because we're really measuring the disinfectant's ability to diffuse from the disc. And that's not the way we use disinfectants. Generally, with a disinfectant, you put it on a rag and then you wipe down a surface. And so we're not using the disinfectant in its property to diffuse through an agar plate. And so that's why this test is not so good at determining which disinfectant is the best one to use against this bacteria, because it's not the way we're using disinfectants. We don't generally have a disinfectant diffusing through something, like a disinfectant diffusing through tissue. Does anyone remember when Donald Trump suggested, I think it was chlorine bleach, that we should give that to patients who have COVID-19 and watch the disinfectant kill off the COVID-19? It might work in killing the COVID-19, but it would also kill the patient, okay? So generally we don't use disinfectants by diffusion. The inhibition zone size of inhibition for an antibiotic is dependent on several factors. It is dependent on the bacteria sensitivity to the organism, but it's also dependent on the growth rate of the bacterium. It's also dependent on the diffusion of the antibiotic through the agar. And Table 6.2 looks at some of the standardized protocols for the Kirby-Bauer test, that when we use the Kirby-Bauer test, you should always follow this protocol to standardize it so that your results on your Kirby-Bauer test will be similar to the results of another person across the world who did the same Kirby-Bauer test. Meaning you should inoculate the nutrient broth from several bacteria colonies, and you grow that bacteria on non-selective media, and the bacteria are no more than 24 hours old in that non-selective media. You then use the turbidity of the growth of the bacteria at 0.5 McFarland units. This is approximately 10 to the 5th bacteria cells per milliliter. You inoculate the bacteria at room temperature on Mueller-Hinton agar plates. We always use Mueller-Hinton agar plates of a pH of between 7.2 and 7.4. That's a mistake there. That depth should be 5 millimeters. And you put the bacteria on using a non-toxic swab, using the spread plate technique to obtain confluent growth. I'll talk more about that in a little bit. And then when you put the antibiotic discs on, the discs should be at least 24 millimeters apart, no more than 12 discs per plate, so 24 millimeters apart there at least. If you get them closer together, they can interact. And then you incubate it in a standard non-CO2 incubator for 16 to 18 hours at 35 degrees. And then you incubate the plates no longer than 15 minutes after adding the antibiotic disc. Okay, so we standardize all those variables so that your Kirby-Bayer test will be similar to somebody else's Kirby-Bayer test. Okay, any question about that? You then measure the inhibition zone size of the zone of inhibition. You measure the diameter using millimeters. Here's an example right there. And usually you take two measurements, so you go this way and then that way, or this way and then that way, and then you take an average if they're different. And this one is showing you the zone of inhibition of Staphylococcus aureus with the antibiotic penicillin. It's probably penicillin G. And the zone of inhibition is 20 millimeters. And then you look that up in a table, the NCCLS table, which is the standard table. If the zone of inhibition is less than or equal to 20 millimeters, then we say that this bacteria is resistant to that antibiotic. If it's greater than 29 millimeters, the zone of inhibition, then we say that that bacteria is sensitive to that antibiotic. And if the zone of inhibition is between that, between 21 and 28 millimeters, we say that the bacteria is intermediate to this antibiotic. And so you have to look on a table for each species and for each antibiotic to determine whether the bacteria is resistant to the antibiotic or sensitive to the antibiotic. And what this table is doing is it's assuming you're following standard Kirby-Bayer procedure, and it's looking at the different sizes and taking into account the diffusion of the antibiotic through the disc, through the agar. Penicillin, I think, diffuses okay, and 20 millimeters is a moderate size. An antibiotic that's much larger and doesn't diffuse very much will have a smaller zone of inhibition, and so its measurements will be less than this. It'll be resistant maybe if it's 10 millimeters or less, and sensitive maybe if it's 20 millimeters or greater in size. Any question about this? So to determine the sensitivity of a bacteria to an antibiotic, you have to measure the zone of inhibition and then look it up in the NCCLS table. And can anyone remember what NCCLS stands for? The National Committee for Clinical Laboratory Studies. All right, any questions about any of that? If not, learn the terms for this lesson. In this laboratory, you're not going to be physically performing any of these activities, but you will be responsible for knowing how to interpret the effectiveness of antibiotics using the Kirby-Bayer disc diffusion test. Watch the video clip on preparing a spread plate, and then review the steps using aseptic technique when preparing a spread plate or bacterial lawn of growth. And that's right here. Inoculate as much of the agar's surface area as possible with the swab, including the edges. You need to do it 3 times. So let's pretend that this is an agar plate, and this is my swab. You dip it in your bacteria, and generally you would, when you're taking it out, you get the excess off by squeezing it, I don't know if you can see that, on the side of the tube. And then you swab everywhere on the plate. Obviously this isn't round. And then you rotate the plate, and you rotate the swab, and you do it again. Going everywhere. I'm just going to do it quickly here to show you. Then you rotate the plate, and you rotate the swab, you do it a 3rd time, and you do it again. You want the bacteria growing everywhere on the plate. That's why we call it a lawn of bacteria. I once had a student which she went, up, da, 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 up, da, 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 and then her plate had a whole bunch of S's for the bacteria growing on it, and very large regions where no bacteria was growing, and she couldn't measure her zone of inhibition. View the clip on preparing a spread plate, or I talked about that, sorry. I forgot I moved there. Anyways, that's how you do it. And then view the videos for evaluating the antibiotic effectiveness, the interpretation, the results for the Kirby-Bayer disc diffusion, and then the virtual lab, if you can get it to run in Flash. This will give you a practice on doing the lab. This program, once you start it, click on the link. It will take some time to load, so give it some time to load. After several minutes, 3 to 5 minutes, if it isn't working or it shows you a message saying that the Flash player isn't going to play because, I don't know what it says, something like Flash has been discontinued, something like that. You try your best, use a different browser to try and get it to run. I got it to run with Google Chrome, I think, when I opened it directly from the page, meaning I had to download the document. I'm going to try and show you this document. Come on. I can't show you. There it is. Download the document onto your computer and then run it from your computer. It did not run for me, the Flash player, when I was trying to run it from Canvas. Okay? So to get the Flash player to run, you may need to download it, the document, to your computer or whatever. And then use the standards required for the Kirby-Bayer susceptibility testing, preparing using a spread plate technique. Antibiotic discs are placed on the surface of the agar and then incubated overnight. And below are the results along with the NCCLS zone standards used for interpretation. Take a look at this, and it's showing you the measurement of the zone of inhibition, the circle, for ampicillin, cephalofithin, gentamicin, and tetracycline. When you're measuring this, you do not measure this right here. I don't know, this is about 9 millimeters. You measure it where the 0 mark is. And if you notice, on the calipers here, this region here isn't measured. And that's why you're measuring at the 0 here. Okay, any question about that? So this would be 15 millimeters for ampicillin. It's not 9 millimeters. Any question about that? And then come down to the table, and let's see, is that E. coli? I assume we're seeing E. coli here. Yes, it's E. coli. So for E. coli, 15 millimeters. For ampicillin is intermediate. Any questions about that? Everyone's good on it. Okay. And then, not here, but in the worksheet, fill in the questions, and I changed this so that you can put in a measurement there. You can actually write in the document. And then measure the zone of inhibition millimeter in your interpretation for question, the first question is question 2. And then answer the questions here for question 3. Wherever there's a dot, that means you should put an answer. And then do the questions in the worksheet, which there's also 7 more questions. And I'll be here till 8:20 to answer any questions. All right? I'll be up and about, but all right. So go ahead and get to it. I'm going to stop the recording. Take that. Somebody put in a chat message. I've told you before, don't put in the chat message, because when I'm showing my screen, I can't see that I have a message. And so I had no idea that somebody had asked a question of me, and I don't use it because I can't see it. If you have a question, you got to ask it. All right? Don't use the chat box because I can't see it.